Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at uncovering some of the secrets of bone cells. So hang around with me throughout this entire video to get all of the relevant background information so that way we can dive into some exciting experimental results. Plus, if you look in the description below, there is some information on how to get your hands on a free NFT. Now, of particular relevance to the topic at hand is that this coming week of October 12th to 20th is Bone and Joint Action Week, which focuses on many different musculoskeletal disorders. The themes and their related activities are designed to raise awareness worldwide about prevention, disease management, and treatment. So to give some context for this, more than half of the American population over the age of 18, 54% as of 2011, are affected by bone and joint conditions. Now, I have a bit of skin in this game because I'm also affected due to some bad genetics and playing contact sports when I was younger. Probably a poor choice, but tons of fun. Anyway, this incidence of musculoskeletal disorders is comparable to the total percentage of Americans living with a chronic lung or heart condition. I would give you more recent numbers, but I had trouble finding anything more recent than 2011, which hopefully means that someone will publish new numbers soon. But a decade ago, musculoskeletal conditions cost an estimated $213 billion in annual treatment, care, and lost wages, accounting for over 5% of GDP. Now, these numbers are still projected to increase by $73 billion up to 2024. Musculoskeletal conditions include back pain, arthritis, traumatic injuries, osteoporosis, spinal deformities, and childhood conditions. And the way that we're going to look at how bone cells function today is by investigating something called osteopetrosis. Now, this is not some fancy spell that gives us unlimited knowledge about bone cells, but that would be incredibly handy. This is a rare genetic condition characterized by reduced bone resorption leading to increased bone density. Now, the most malignant form of osteopetrosis leads to death within the first decade of life if left untreated, and results from an autosomal recessive mutation, and affects 1 in every 20 to 250,000 newborns worldwide. Autosomal dominant mutations take a little bit longer to diagnose, being recognized in late childhood and adulthood, and result in a milder, non-lethal form of the disease that still affects osteoclast numbers or function and affects 1 in every 200,000 individuals. Now, this is what normal bone development looks like on a radiograph. You've probably seen them before. These bones are continuously maintained by multinucleated osteoclasts breaking down and resorbing bone, and osteoblasts making new bone. All forms of osteopetrosis share the hallmarks of increased bone density and thickness on radiographs, and are a result of osteoclast absence or malfunction where old bone is not being broken down, while the osteoblasts continue to make new bone. Now, there are many clinical features of osteopetrosis, namely frequent fractures due to a loss of elasticity and high rigidity of the bone, macrocephaly and frontal bossing due to bone thickening, cranial palsy, which results in hearing and visual impairments alongside facial palsy, malformed sinuses, cranial nerve deficits, tooth decay, poor dentition, and other features such as scoliosis and anemia. Osteopetrosis comprises a clinically and genetically heterogeneous group of conditions that can have mutations in one of many genes in humans, such as TNF-RSF11A. And here are some more genes that can target functions of osteoclasts, like mutation of cathepsin K. And there are homologs in mice, like the mutations in CSF1R, which are used to study this disease. Osteopetrosis can be treated by bone marrow transplants, if caught early enough in infants. But unfortunately, there is no definitive cure as of yet. Now, most of my other content has been dealing with the immune system. So why am I now talking about bone health? There are many reasons for that. 1, it's Bone and Joint Health Action Week. 2, spooky season. 3, I think it's just neat to learn about related topics. And 4, bones and the immune system are actually intrinsically related. First off, during fetal development, which in mice lasts 21 days, compared to humans, which lasts about 280 days, tissue-resident macrophages arise from erythromyeloid progenitor cells in the fetal liver and travel to many different tissues throughout the body to function and act as sentinels which help deal with infections or injuries immediately at the sites. Hematopoietic stem cells develop from the bone marrow after birth and circulate throughout the body and help replenish tissue-resident macrophages. Now, these hematopoietic stem cells have the ability to develop into any of the other immune cells depending on what type of genes they express and the factors they're exposed to. They can turn into cells like T cells, B cells, and macrophages, which are all essential for complete immune function. An example of how they differentiate is with monocytes. the precursors of macrophages. They differentiate from granulocyte monocyte progenitor cells in the presence of MCSF, and osteoclasts develop from the progenitor cells or monocytes in the presence of rank ligand and CSF1 by recognizing them with expressed receptors. With this information in the back of our heads, I'm next going to highlight why investigating rare diseases and understanding their mechanisms of action are important. Even though osteopetrosis is rare, it is still treatable, and the option should be available to treat it. And in this case, it is through hematopoietic stem cell transplantation. Now, this is a very difficult procedure that requires irradiation or chemotherapy, which increases the risk of infections. And those who have this disease usually also have severe complications. Thus, the 6-year survival rate is just under half. So we need to find a better solution. Additionally, by learning about how osteopetrosis functions and the cells that are involved in it, we can apply this knowledge to help find a cure and learn more about other diseases that use the same cells. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today, called Developmental Origin, Functional Maintenance, and Genetic Rescue of Osteoclasts by Jacome Galarza et al. From the Sloan Kettering Institute in New York, USA. And in this paper, the authors use mouse models to investigate the depths of bone cell function and development. Now, to probe the origin of osteoclasts, the authors manipulated genes that are specific to individual cell types. They mutated CSF1R, which is expressed during macrophage production, differentiation, and for function, and TNF-RSF11A, which is a gene that is required for osteoclast development. Osteoclasts play an important role in the development of teeth and bones, and the authors knocked out the CSF1R or the TNF-RSF11A genes in macrophages. And they found that this resulted in a lack of tooth production and deformation of bones in the skull and shortening of long bones. The cells that they were looking at were present here. And to ask whether osteoclast differentiation requires expression of TNF-RSF11A and CSF1R, the authors targeted those genes in hematopoietic stem cells and in lymphoid cells such as T and B cells. FLT3 is expressed during normal hematopoietic stem cell differentiation, and VAV is expressed during B cell and T cell development and activation. So by knocking out these macrophage and osteoclast genes in hematopoietic stem cells, T cells, and B cells, the authors found, unsurprisingly, that normal development of teeth, bone marrow, bone cellularity, and osteoclast numbers were present when these cells were knocked out of T cells, as they're not macrophages. But surprisingly, they found normal development of teeth and bones when these genes were knocked out of hematopoietic stem cells. However, these mice lost osteoclasts over time and had lower levels of hematopoietic stem cells in long bones. Thus, these data suggest that osteoclast development, tooth eruption, and development of bone and bone marrow cavity require precursors that are independent from the hematopoietic stem cell lineage. Now, the authors hypothesized that perhaps the precursors of osteoclasts may develop specifically in the erythromyeloid progenitor lineage of resident macrophages, independent of normal hematopoiesis. When macrophages expressed yellow fluorescent protein, shown in green, the osteoclasts retained fluorescence throughout life. However, when hematopoietic stem cells expressed YFP, the osteoclasts did not have fluorescence before birth, even though these cells were present in the bone marrow. But after birth, both cell types got incorporated into osteoclasts. This indicates that fetal osteoclasts arise from erythromyeloid progenitors in bone development centers, and that postnatal contribution of hematopoietic stem cells is important for optimal osteoclast maintenance. The authors then investigated whether erythromyeloid progenitor cells are required for bone development. So TNF-RSF11A is expressed in osteoclasts, but its expression is also a hallmark of erythromyeloid progenitor-derived macrophage precursors that colonize the developing embryo. So by knocking out the macrophage development gene out of these cells, they could see the effects of erythromyeloid progenitor cells on osteoclast development. The authors hypothesized that this mutation would recapitulate the macrophage deficiency observed previously with the CSF1R knockout mice, while leaving hematopoietic stem cells unaffected. So once again, here's what a normal mouse skeleton looks like. But the authors used two mouse strains called Koba and Wask, which had TNF-RSF11A knocked in, which highly expresses in fetal macrophages, but not at all in hematopoietic stem cells. And they found when knocking out CSF1R in these mice, that they lacked tissue macrophages in other tissues like the brain and had no tooth eruption. Now, these mice still had stunted bone growth, but had colonization of the long bones by osteoclasts and hematopoietic stem cells, thus indicating that circulating macrophages can enter into and recolonize bone marrow. Now, the authors next asked What contributions do hematopoietic stem cells have to osteoclast maintenance? To do this, they utilize a technique known as parabiosis. Parabiosis is the surgical union of two organisms developing a single shared physiological system. Now, this can prove that a secreted factor in one animal impacts the physiology of a second animal through their shared circulatory system. So, to probe the mechanisms that underlie the contributions of macrophages to osteoclasts, they took two mice, one that expresses YFP on its macrophages, and another that has TD tomato on its macrophages. And to determine the lifespan and the dynamics of osteoclasts in vivo, they performed time course parabiosis experiments. After 4 to 8 weeks of shared blood circulation, all osteoclasts lining the bone marrow expressed both YFP and TD tomato. I want to take a moment here and just say, aren't these colors stunning? So good. They also saw a gradual incorporation of both cell signals into the osteoclasts, which had an average of 5 nuclei. And these colors were not seen together in any other cell type. And this fluorescent intensity decreased after separation, but was still retained in many of the osteoclasts. Now, with a small-scale experiment, the authors found that with one round of treatment after 72 hours, one monocytic nuclei got incorporated into the osteoclasts, which is consistent that individual nuclei get replaced about every 2 months. Now, these data suggest that individual osteoclast syncytia are long-lived, but acquire new nuclei one at a time every 4 to 8 weeks from circulating blood cells. It therefore takes more than 6 months to renew all 5 nuclei in a single osteoclast. In a related condition of adult-onset osteopetrosis, where cathepsin K is mutated, also known as pycnodysostoses, the authors wanted to know if this phenotype can be rescued. So when these mice were paired through parabiosis with heterogeneous or double-positive mice for cathepsin K, their bone density was decreased, indicating functioning osteoclasts. And to test whether expression of donor-derived genes by recipient osteoclasts results from the fusion with monocytic cells in the blood, the authors performed IV injection of monocytes expressing TD tomato into YFP-expressing mice. And this lovely picture here is what they saw after 60 days. They found that about 20 to 40% of osteoclasts uptake monocytic cell genetic material at 1 and 8 weeks after treatment. And they also found that the transfusion of monocytic cells can rescue the osteopetrosis phenotype, and that the injected cells expressing YFP were found in the bone marrow cavity and nowhere else. This indicated that there was a lack of hematopoietic stem cell engraftment and thus no lasting changes to the immune cells of the mice. Now, to summarize everything that we just talked about, the authors of this paper found that a lack of erythromyeloid progenitor cells causes osteopetrosis, and that hematopoietic stem cells get incorporated into osteoclasts in the bone marrow after birth, and are not essential for the osteopetrotic phenotype seen. They also found that monocytes can share their genetic information with osteoclasts after birth, and that the sharing of this genetic material can reverse the symptoms of osteopetrosis. Now, not only is this information just interesting to learn about, but it is also significant because it shows that osteoclasts are originating from erythromyeloid progenitor cells and are essential for normal bone development. Timely replacement of erythromyeloid progenitor cell-derived osteoclasts by transfusion with monocytic cells rescues bone development. The authors also showed that osteoclasts can be targeted for gene transfer and cell therapy based on transfusion of monocytes to modulate osteoclast activity. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions of my own when reviewing this information. Now, my first question is, how easy is it to create monocytes from pluripotent stem cells for treatment? Because cells need to be HLA matched so that the immune system does not reject them. So is this an easy alternative, or do you need to get cells from a compatible donor like a sibling? My second question is, how long will this treatment last? It seems to last a while in mice, but in humans, will it last months, years, decades? My third question has to do with whether this information can be used to treat other bone density diseases, like osteoporosis, which can involve increased osteoclast function. And my final question is, what sort of ideas popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there's any topics that you're interested in hearing about in the future. Ultimately, I hope that you learned something, but more importantly, you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I will see you next time.